let's move to the next topic, post-fertilization structure and events. In the previous class, we discussed about process of fertilization in angiosperm. Now, after fertilization, there are some changes taking place in the parts of the flower. What is meant by post-fertilization changes? Today's topic is post-fertilization structure and events. After fertilization, there are number of changes takes place within the flower. What are the changes taking place? Let's study in detail. The floral parts, there are 4 different whorls like calyx, corolla, androecium and gynoecium. Among this, the calyx is made up of sepal, corolla contains petal, and androecium there are stamen, and gynoecium, that is pistil, which consists of stigma, style and ovary. So, these all parts, there are number of changes taking place. After fertilization, this sepal, petal, stamen, stigma, style, these all parts wither and fall off. As soon as the fertilization gets over, fertilization is the process of fusing gametes, fusion of male and female gametes. So, among this 4 whorls, this androecium and gynoecium, as you know, this androecium and gynoecium are the essential whorls. Remaining whorls, the calyx, corolla, these are help this flower, help the agents to pollination. For pollinating agents, this calyx, corolla, this parts helps. Also, this calyx which protects before fertilization. After fertilization, these all parts wither and fall. The sepal, petal, stamen, stigma, style. Now, this ovary becomes fruit, and the ovule becomes seed. Then, if you see the structure of this ovule, in the ovule, structure of ovule, already you learned what are the structure of ovule. There is a stalk of ovule that's called as funicle or funiculus. So, this funicle is a stalk of the ovule, become the stalk of the seed. And the integuments, the ovule which is made up of, which is surrounded by outer and inner integuments. These integuments becomes the seed coat. The outer integument, there are 2 integuments, outer and inner integument. This outer integument becomes the outer seed coat and the inner integument becomes the inner seed coat. You know the seed which is covered by 2 different types of coats, outer seed coat and inner seed coat, which is derived from this integuments. Then there is a region called micropile. This micropile of the ovule which becomes the micropile of the seed, which facilitate the exchange of oxygen as well as water. So, gaseous exchange and water exchange takes place due to this micropile. The micropile of the ovule which becomes the micropile of the seed. So, these all changes are known as post-fertilization changes. So, these all the changes occurs in the flower. That event is known as post-fertilization structure and event. Now, next part which develops after fertilization is endosperm formation. Endosperm, and the next process is embryogenesis. So, before forming the embryo, there is a structure called endosperm. The next process is called as formation of endosperm. How this endosperm develops? This endosperm develops from primary endosperm nucleus, PEN. PEN. You just for the better understand, you remember this as PEN. PEN, primary endosperm nucleus. The primary endosperm nucleus undergo division and it becomes endosperm. The endosperm is formed in most of the angiosperms. This primary endosperm nucleus, how this primary endosperm nucleus form, in the previous class we discussed very clear. There is the fusion of gametes, it forms the zygote. There are two different fusion taking place, syngamy and double fertilization. After the triple fusion, this primary endosperm nucleus is the result of triple fusion. How it forms? Once again, let's see that. This is the pollen grain, pollen tube. From the pollen, when the pollen germinates, it forms as the pollen tube. This pollen tube carries two male gamete along with the vegetative tube nucleus. This is the vegetative tube nucleus. These two are male gametes. When you see the structure of embryo sac, this is the embryo sac, which consists of synergids, antipodals and polar nuclei. Here there is two polar nuclei. At first there are two polar nuclei. Now what is meant by triple fusion? This, let's see this, here outer integuments are present. This is embryo sac. Embryo sac. Now this structure, there is a fusion of this male gametes. One of the male gamete which fuses with the egg and forms zygote. Another male gamete, this gamete fuses with the secondary nucleus. So this one fused through the secondary nucleus. How the secondary nucleus form? Already there are two polar nuclei. This is known as polar nuclei. These two nucleus fused once and becomes secondary nucleus. Now this is secondary nucleus. This male gamete fuses with this secondary nucleus. So it is N male gamete plus already it is secondary nucleus. Two polar nuclei fused together. So it becomes 2N. So now it becomes 3N. This 3N is the primary endosperm nucleus. So as the result of this, after the triple fusion, there is the formation of endosperm. Here is the endosperm. Inside the embryo develops. This is endosperm, inside embryo develops. So how this endosperm is formed? From the primary endosperm nucleus. This is the result of triple fusion. Hope you understand this clear. Triple fusion. This male gamete fused with the secondary nucleus. So 3N is present here and it becomes the endosperm and inside embryo develops. Based on the nature of development, there are three different types of endosperm, and the function of endosperm is this endosperm nourishes the developing embryo. Embryo is developing within this endosperm. Nutrition given by this endosperm this endosperm nourishes the developing embryo. So what are the different types of endosperm? The next question is types of endosperm. Based on the development, there are three different types of endosperm, namely nuclear endosperm. The first type of endosperm is called nuclear endosperm. Second type is cellular endosperm. Third type is Helobial endosperm. Apart from this, there is another type of endosperm called Ruminate endosperm. So basically there are three different types of endosperms based on the nature of this development. Let's see one by one. Nuclear endosperm. Look at this diagram. This is the development of endosperm, nuclear endosperm. Now this primary endosperm nucleus, this is the primary endosperm nucleus, or you just remember the word as pen. Primary endosperm nucleus. This is the primary endosperm nucleus. The primary endosperm nucleus divides into two. Primary endosperm nucleus divides into two without wall formation. In this diagram, you just observe there is no formation of wall. Wall formation is absent. So, primary endosperm nucleus without wall formation, it divides into two. The subsequent division takes place, free nuclear division. So, these two cells subsequently it divides. Subsequent division is further division. Further this cell undergo division. There is no wall formation. So, this type of division is called free nuclear division. The nucleus is freely on the cytoplasm. So, free nuclear division, all the nucleus are free without wall formation. This type of development, now this is the endosperm. The primary endosperm nucleus becomes the endosperm, formation of endosperm. In the later stage of this, either it is free nuclear or later it produces a wall like this. This type of development is known as nuclear endosperm without wall formation. So, the difference among nuclear endosperm and cellular endosperm is wall formation. In nuclear endosperm, there is no wall formation, so that this endosperm, all these nucleus are free nuclear. Next type of division is called, next type of endosperm is known as cellular endosperm. In cellular endosperm, here is the primary endosperm nucleus, PEN, primary endosperm nucleus. This primary endosperm nucleus undergo division. Immediately after division, immediately it divides, it forms, followed by the wall formation. So, here is the wall, cell wall. The cell wall divides the two nucleus, and subsequent division also followed by cellular wall formation. Here look at this diagram, there is a difference. Here there is no cell wall formation, but in cellular endosperm development there is a wall formation. Subsequent division with wall formation. So, finally it becomes the endosperm. All the endosperms or all the nucleus, there will be a wall formation. This is the main difference among nuclear endosperm and cellular endosperm. Next type of endosperm is known as Helobial endosperm. Look at this diagram. In the primary endosperm nucleus, this is the primary endosperm nucleus. In this Helobial type, the primary endosperm nucleus, see here the primary endosperm nucleus is present at the center or middle part. But in this, the primary endosperm nucleus moves towards the chalazial region. This is the micropilar end, this is the chalazial end. So, it moves to the lower part or the chalazial end, and it divides into two. It divides, the primary endosperm nucleus divides into two, and it produces two different chambers. Look at this diagram. There are two chambers. The upper part, that is towards the micropilar chamber, is very large, and towards the chalazial chamber it is very small. So, this is the micropilar chamber, this is the chalazial chamber. Now this micropile chamber nucleus which divides free nuclei, which undergo free nuclear division without wall formation, the micropile nucleus, from the micropile chamber there is a nucleus. This nucleus divides freely as a free nucleus. It forms as a free nucleus. And the chalacer chamber which forms as a wall layer. So this is the nature of the division, and it becomes the endosperm. So endosperm development taking place. So here there are two different chambers, micropile chamber and chalacer chamber. So when they ask the question, you differentiate these three. What is the difference among nuclear endosperm development, cellular endosperm and the helobial endosperm? Then apart from this, there is a type of endosperm that is called ruminate endosperm. In ruminate endosperm, there is a irregular or unevenness in the surface of the endosperm. That type of endosperm is called ruminate endosperm. So at first there is a endosperm formation. Now what's the function of this endosperm? The function of the endosperm is this endosperm. Now endosperm is formed as a result of triple fusion. Now this endosperm helps the developing embryo to nutritive. So it nourishes the developing embryo. That's the main function of this endosperm. And in most of the angiosperms, the endosperm forms before the development of embryo. So zygote divides after the development of endosperm in most of the angiosperms. And this endosperm, it helps to the precise development of this embryo. So the mode of embryo development is recognized, or it is recognized by this endosperm. So it nourishes the developing embryo. The zygote divides after the development of endosperm. So first endosperm develops, later the zygote formation. Based on this, so sometimes in certain species or some species, based on this type of endosperm, there are two. How it is? The endosperm, when it is nourishes the embryo, sometimes the endosperm completely consumes. When it is consumed completely, the endosperm, there will not be a endosperm. So they are called as X-albuminous seed or non-endospermous seed. So the seed is called non-endospermous or X-albuminous. Why it is named like this? When the embryo develops, it consumes all the endosperms. So there is no endosperm, non-endosperm, it is non-endospermous. But if endosperm is present, that is called as endospermous embryo. So endospermous X-albuminous or albuminous seed. If endosperm is present, it is endospermous seed. If endosperm is absent, when it is consumed completely, it is called non-endospermous seed. So based on this, there are two different types of seed. Now next process is called as, then another structure is there, development of hastoria, endospermal hastoria. Now basically this hastoria will develop from this Helobial endosperm. In the Helobial endosperm, this chalacer chamber, this is the chamber, when it divides, at the time of primary endosperm nucleus divides, there are two different chambers develop, micropile chamber and the chalacer chamber. In Helobial endosperm, this chalacer chamber either acts as a hastorial cell, hastoria, or in cellular and nuclear endosperm, the micropile region or the chalacer region or both the regions which develops into the which produces the hastoria. So, hastoria develops from the endosperm. Now, next process is known as embryogenesis. So, it is also very important question, embryogenesis, development of embryo. Let us see how the embryo develops in the dicot seed. This is the development of these diagrams which illustrate the development of dicot embryo. As I told you earlier, here the zygote, this is the zygote. This zygote divides after the development of endosperm. Before this zygote develops, endosperm forms. Now, this zygote contains a nucleus. This zygote which divides as a result of fusion, as a result of syngamy, that means fusion of male gamete, this male gamete with the egg cell in the synergy. Among this synergy, this male gamete fused to the egg and forms zygote. Now, this is the result of syngamy. This process is known as syngamy, fusion of male gamete with egg. Male gamete fuses to the egg and forms zygote is called syngamy. So, this is the result of syngamy, zygote. The zygote undergo division. So, it divides into two. There is a transverse division taking place. This type of division is called transverse division. The zygote undergo transverse division and it produces two different cells, the upper cell and the basal cell. The upper cell is called as the terminal cell, upper or terminal cell, and the lower or basal cell. So, zygote undergo division, transverse division, and becomes terminal cell and the basal cell. So, now this cell is called as two-celled proembryo. So, zygote which divides into two, and the name of the division is called, now this stage is called as two-celled proembryo. It forms as a two-celled proembryo. Now, next division taking place in the terminal cell. The terminal cell divides vertically. There is a vertical division takes place in the terminal cell. So, which becomes two, and it produces this, there are two different cells and the basal cell which divides transversely. So, two division taking place, a vertical division and a transverse division. The terminal cell or the upper cell undergo vertical division and the basal cell which undergo transverse division. Now, this produces four different cells. Now, this stage is called as four-celled proembryo. Now, next stage is four-celled. First two-cell stage, the proembryo is two-cell. Now, the proembryo is become four-cell. Now, this cell, the upper part or the terminal region which undergo transverse division at right angle to the earlier, and it produces a four-celled stage, the terminal cell alone, which forms a four-celled stage which is called as quadrant. So, now this stage is known as globular embryo, globular stage. So, here it divides this upper cell, terminal cell which divides into four. These two cells now becomes four cell. Now, this stage is called as quadrant stage. So, this quadrant, now this quadrant cells, four cell which undergo division and it produces 8-cell stage. So, 8-cell stage which is known as octant. So, first quadrant, from the quadrant it produces octant. There are 8 different cells. These 8 different cells are arranged as 2 tiers. The 4 cells are arranged in the upper tier and the remaining 4 arranged in the lower tier. So, in the upper octant stage, octant cell, upper octant cell, the 4 tiers of cells in the upper region is known as epibasal cell, and the lower 4 tiers of cells are known as hypobasal, epibasal and hypobasal. So, first 2-cell stage, then 4-cell stage, now quadrant stage. Now, quadrant stage become octant. Octant, there are 8-cell stage. These 8 cells are arranged in 4 and 4, upper 4, lower 4. Upper 4 is known as, they are arranged as a tiers. This upper 4 tier of cells is called as epibasal, and the lower tiers of cells are known as hypobasal. Now, this 8-cell stage or the octant stage, which undergo division, and it produces 16 stage. So, 2, 4, then 8, now 16-celled stage. So, this 16-celled stage also arranged as 2 tiers of cells. Our upper region there are 8 cells are arranged, lower tiers there are another 8 cells. Likewise, it produces 16-celled stage. Now, this 16-celled stage later, now in this stage, quadrant stage, it becomes globular. Later it becomes different shapes, the heart shaped, and it becomes the mature stage. During this development, so when this 16-celled are formed, the 16 cells again undergo periclinal division and undergo anticlinal division, and it produces and it becomes the mature embryo. The mature embryo which forms the epidermis, cortex, and the stela region. So, these 3 regions are derived from this 16-celled stage. The 16-celled stage undergo various division periclinally and anticlinally. During this type of division, here are the basal cell. This basal cell, now upper cells, it becomes globular stage, quadrant, octant, and 16-cell stage, and it produces epidermis, cortex, and the stela region. Now, the basal cell of this 4-celled proembryo, this basal cell which undergo division, transverse division, and it forms 6 to 10 celled stage. It produces 6 to 10 stage. Now, this forms as a suspensor. The name of this part, the basal cell, upper part which is called quadrant, but the basal cell which undergo division, and it forms 6 to 10 cell, and they are called as suspensor. So, these all together called, these all cells, 6 to 10 cell stage is suspensor. And this cell of the suspensor, the first cell of the suspensor which elongates, and it becomes the haustorium. The first cell of this suspensor region, these are the suspensor region. So, this region, it elongates and becomes haustorium. Likewise, this cell, it is known as hypophysis. This is the hypophysis, this is the haustorium. So, 6 to 10 celled stage, this cell elongates and forms the haustorium, and this cell is known as hypophysis. Then the next division taking place in the hypophysis. So, when the division takes place in the hypophysis, now this hypophysis becomes, here is the hypophysis and this is the suspensor, and there will be a embryonal mass develops. So, next further divisions taking place, and it produces the globular stage become heart shape. Now, suspensor is there, and this suspensor, and it forms the embryonal mass is present here. This is the hypophysis, and the shape of this stage is called as heart shape stage. Then from this heart shape, there is a horseshoe shaped stage. This stage is called as horseshoe shaped stage, and later development, it produces the root cap and the radicle. Here there are further division taking place and forms. This is the suspensor, and radicle and root cap is formed, and the mature embryo which consists of radicle, cotyledon, and plumule. So, in the embryo which produces radicle, cotyledon, and the plumule is present. So, in the mature embryo which consists of radicle, cotyledon, and plumule. This is the development of the dicot seed. So, embryogenesis, the name of the process is called as embryogenesis, development of embryo. I hope you understand it clear. Let's recall once again what are the questions you learned today. The first question is post fertilization changes. What are the events and structure develops? So, here ovary becomes seed, ovary becomes fruit and the ovule becomes seed. Finally, after post fertilization changes, remaining parts, the petal, sepal, stamen, stigma, these all parts wither and fall off. Next question is types of endosperm. So, endosperm formation, types of endosperm. There are 3 different types of endosperm: nuclear endosperm, cellular endosperm, helobial. In addition, there is an endosperm called ruminate endosperm. Then endosperm development. 3 types of endosperm development. What's the function of endosperm and how the haustoria is formed. Finally, there is a question embryogenesis. Learn all these questions before the next class. Thank you.